So I have been in UC Irvine for almost more than 20 years now. So my lab is actually focusing on the development of the proteomics-based approach to study protein complexes, especially those in the ubiquitin proteasome system. And although the proteomics analysis can provide all aspects of proteins, so we have focused on to study the protein-protein interactions. So from all the previous speakers' talk, you already see all the great examples of how important the protein-protein interactions are. I just want to emphasize that protein-protein interactions are fundamental to the protein complex formation, and also that it's essential to the understanding of protein structure and the functions, and as well as protein network biology, which are important to understand the phenotypic responses of all different types of cells. And also that the protein-protein interactions can regulate multiple cellular processes, and including the examples have been shown for protein degradation. And the protein-protein interactions are also defined by different parameters. They can be defined by protein strength, such as strong and weak, and also duration, the stable and the transient, and also whether the interactions are dynamic or not. And also that the protein-protein interactions, they are spatially and temporally controlled. And in addition, that due to different extracellular or intracellular signaling perturbation, the protein-protein interactions can be changed. And if you have different types of post-translational modifications, that could also change the protein-protein interaction. And protein-protein interactions has also been shown that they are cell-specific or tissue-specific, and these are important for their biological functions. So there are many different types of protein-protein interactions methods that have been developed. So for mass spectrometry-based approach, one of the methods is affinity purification mass spectrometry. This method has become, the method of choice to study the protein complexes. And this approach involves, the expression of a tagged bait, and then after cell lysis, then the bait and the prey will be purified through affinity resin binding, and after elution, it was subjected to mass spec analysis and to generate the composition of the protein complexes and also the interactome. And this approach has really been powerful for studying systems-wide protein-protein interaction analysis, especially now there's more than the 4,000 proteins have been tagged for studying this kind of the networks from different organisms. Why this approach is very effective, it actually has the sensitivity showing the speed, very versatile, and also it has the scope of analysis for different organisms. But every method has its pros and cons. So for this one, it has some limitations. One is that it could form the non-physiological interaction because the interactions are captured after cell lysis, and also can lose the weak and transient interaction due to the cell lysis and also extensive washing steps. And because of any, for any types of resin-based affinity purification, you will have a non-specific background. And also for this approach, although you can identify the composition of the protein complexes, but it doesn't have the information to allow you to determine direct interactors or indirect interactors. And because it requires the tagged bait, so it needs the cell engineering. So that would eliminate its application to clinical samples. So recently, that in order to eliminate some of the disadvantages in the APMS studies, the proximity labeling-based approach has been developed. So as shown here, there are three typical ways to establish the proximity labeling, and these one could use the peroxidase or use the biotin ligase, or use the photocatalyst to try to label the protein interactors within the proximity, which normally typically is 10 to the 20 nanometer. And these approaches have been shown in different organisms and actually can be used for in vivo protein-protein interaction mapping. And also it has been shown to map weak and transient interactions. Why it's very effective and widely accepted by the field. And this approach also has some limitations. One is that it actually has a large background coming from the affinity purification as well as the labeling background. And also it requires, most of the time, requires the cell engineering as well, and also cannot distinguish the direct from indirect interactors. And another thing is that the labeling specificity and sensitivity actually, needs to be further improved. So alternative strategy, try to map protein-protein interaction in my lab. We try to focus using the cross-linking mass spectrometry. And cross-linking mass spectrometry actually is a mass spec-based structure tool for studying native protein interactions. It can capture the interactions through the formation of a covalent bond between proximal amino acids. And this proximity is defined by a specific cross-linker. And this cross-linker is composed of two reactive groups, and then in between is connected by a spacer region. So typically, this region is 7 to 15 angstrom. So the distance between the amino acids actually is much shorter compared to proximity labeling. And there are different reactive groups can be added and to form the covalent bonds between the protein interactors. So why the cross-linking mass spec is special? Because that the cross-linking can capture the protein, endogenous protein-protein interactions in the cellular environment without cell engineering. And it can detect the identity and the context of a protein-protein interaction concurrently. And also, the information generated by the identification of a cross-linked peptide can be used to validate or refine existing structures and also assisting de novo structural modeling. And in addition, it can be used to define protein conformational changes, and also that this method enables system-wide three-dimensional protein-protein interaction mapping. So for us, when we first started, we were trying to use cross-linking mass spectrometry to capture the transient and weak interaction, and especially this is through the study of the proteasome complexes. At the time when we tried to do so, that affinity purification mass spectrometry has been used, but they often only capture the stable interaction. So in order to freeze the interaction prior to cell lysis, we have incorporated the in vivo cross-linking. So here we developed a method called QTAX, indicating its quantitative analysis of tandem affinity purified in vivo cross-linked protein complexes. So here we had expressed a protein with HB tagged and then compare with the control, and this after cross-linking, in-cell cross-linking, then we did a denatured cell lysis and mixed together, and through tandem affinity purification under fully denatured condition and the quantitative mass spectrometry, we were able to identify the specific interactors. So we were able to capture a variety of proteasome interaction when we first started, and not only the stable, but also the weak and transient interaction. So this technique has been applied to different systems, and recently we also applied it to a plant system to capture signaling complexes. And so when we first started, we started this in the yeast system, and later we expressed the HB tagged as RPN11 subunit, which is a proteasome subunit in mammalian system, and we have found that we can modulate the in vivo cross-linking conditions to allow us to purify not only denatured condition, but also native condition to preserve the interaction, but also preserve the structure and their functionality. So these are different approaches to allow you to capture the interaction and for different purposes. So I don't want to bore you with all the details for cross-linking mass spectrometry, but I wanted to tell you the basis for the cross-linking mass spectrometry for structural analysis. And this is through the identification of the cross-linked peptides. And so when you have a cross-linked protein after enzymatic digestion, it will result into different types of peptides. You can see here's a dead end, meaning one end, coupled with the amino acid, but the other end is actually hydrolyzed. And then you also have a cross-link formed between one peptide or the two different peptides or multiple peptides. So in here, these type of cross-link peptide called interlink peptide, they are most structural informative. And because we have all these different types of products forming here, and then the end of the products for analysis is heterogeneous, and often these interlinked peptides, they are very low abundant. So in mass spectrometry, when you try to analyze cross-linked peptide, and here shows the example of interlinked peptides. We will fragment the peptides and break the peptide backbone. So in here, you can see that you will generate different fragments coming from either alpha or beta peptides or both alpha and beta peptides. So it makes the spectra very complicated compared to the conventional database searching of the linear peptides. And although that the bioinformatics tools have been developed to try to analyze this kind of data, but in general, when you have a larger amount of samples, a complex sample, it is difficult. So in order to facilitate or advance the cross-linking mass spectrometry, we have to try to develop a simplified strategy, which is through the development of sulfoxide-containing MS cleavable cross-linker, and for simplified MS analysis and identification of the cross-linked peptides. So the first one we developed is called diisocyanamide sulfoxide, and in short, it's called DSSO. So in here, the key is the central sulfoxide introducing a MS cleavable bond, and this bond is much weaker than the peptide backbone. So during the mass spec analysis in the MS1 stage, you actually detect the parent ion. In MS2 stage, you can cleave the cross-linked peptide into individual peptides, and then you can do the stage 3 mass spec, and each individual peptides can be fragmented, and this spectrum much simpler, and you can use conventional database searching tool to analyze it. So DSSO, as shown by Cheetah, used in his study, here consists of 2 NH ester targeting the primary amine group, and especially the lysine for cross-linking. So in order to analyze this, I wanted to show this slide for you to understand the information we generated. So for here, we use the LC-MSN to acquire the data, and then to use the conventional database searching tool to search the linear peptides. Because interlinked peptides, actually, if you use to do the database searching, in general, you will have actually expansion of the database searching space. But with this type of MS cleavable reagent, and we can just search the linear peptides, is much simpler with higher accuracy. So once we identify those cross-linked peptides, and then we use integration of all the data, and then we can identify the cross-links. For this example is K2K linkages, and then these linkages allow us to determine the contact sites. So with this information in hand, we actually can derive the site-specific PPIs of protein complexes. Here, I use the proteasome as an example. So when we first started when we published this work in 2012, before we started the work, actually there's no atomic resolution structure of 26S proteasome available. At the time when I tried to publish this work, that actually 2 papers coming out with the cryo-EM structure. But nonetheless, the cross-linking data we obtained matched very well with the existing structure. But in addition, we were able to map the proteins which are a very small subunit in the 26S proteasome, which the information was not revealed by cryo-EM because of the low density of those proteins, and also including those dynamic interactions. So then initially we studied the yeast 26S proteasome, and then we extended to study the human 26S proteasome. So in here, we did the in-cell and in vitro cross-linking, and not only we generated the comprehensive interaction network, but also we were able to capture the interactors, the regulators, their interaction with the 26S proteasome, including the different regulators such as UBL-CP1 as a dephosphorates of the 26S proteasome. And then recently, in collaboration with John Hanna's group, we actually kind of complement the cryo-EM structure of East 20S proteasome and to localize one of the proteasome inhibitor, FUB1, and their location in the 20S proteasome. So in addition to map the specific site and to understand the protein context, we also can use the cross-linking information to assist the integrative structure modeling. Because for the cross-linker, there's a distance restraint, and this distance restraint is defined by specific cross-linker. And from that, we were able to show that we can use cross-linking information alone to derive the connectivity of the subunits within a complex. And we can generate the localization of the regulators in the proteasome complex. And we can also model the ECM29, which is a regulator of the 26S proteasome under oxidation stress, and then to show its localization and also to understand its regulation of the stress-mediated triggered disassembly of the 26S proteasome. And then use this information, we can generate the model of the COP9 signalosome. And so all this information allow us to not only generate the interaction, but also the structural aspect. So in coupling with quantitative mass spectrometry, we actually can actually determine the relative abundance of the cross-linked peptide, and use this information, we can determine the conformational states differences. For example, here, in collaboration with Lin's lab, we actually used a stable isotope-coded cross-linker to determine the nadation-dependent conformational change of the choline ring ligases. And then use the conventional sialic-based quantitation, we were able to determine the stress-induced conformational change of the 26S proteasome and to derive intermediates of the 26S proteasome structure and correlates with its stress-associated disassembly. And in addition to the comparison of different conformational states, we can also try to do the targeted quantitation. So this is very useful for I often call this as like a structural MS Western, because using the cross-linked peptides, you can specifically to try to dissect regions of protein complexes to understand their structure changes under different conditions. So we use the MS cleavable reagents and then through the fragments, and then these fragments allow us to do the quantitation. And specifically for one of the example is that when we studied the COP9 signalosome, the conventional one is only 8 subunits, and later on, realized there's another subunits present inside. So use the targeted quantitation, we are able to modulate, to determine the differences between the interactions between the two subunits, especially the CSN2 and the CSN3, and then to understand the conformational changes induced by the new subunit CSN9. And then we can also use these to determine the phosphorylation-dependent structural changes within the 26S proteasome, and then different different phosphorylation present in the 26S proteasome induces the conformational change within the 19S, also between the 19S and the 20S. So basically that this type of targeted quantitative mass spectrometry allow you to actually enhance the sensitivity, and this is could be very useful for later on when you try to do the drug-targeted protein-protein interactions. And so in addition, we also found that using multi-chemistry cross-linking mass spec, we can expect the PPI coverage and also increase the precision of integrative structure models. So in here, in addition to DSSO targeting lysine group, we also developed a DHSO targeting acidic group and the BMSO targeting cysteine group. So here, use the COP9 signalosome as an example here. This is 1 to 8. COP9 signalosome here is 1 to 9, and then the interaction between the 9 and the other subunits is only retrieved by the DHSO, which is the linker targeting the acidic group. And so using all these different cross-linkers, we were able to increase the precision of the integrative structure modeling, as you can see from here. And if you have the only one, we generate the model with 29 angstrom precision, and with the 2 cross-linker, we has a 24 angstrom, and then with the 3 linker together, we can improve the precision to 16 angstrom. So basically, that extends the coverage and improves the model precision. So in addition to specific cross-linking analysis, we'll also try to study the protein-protein interactions on the global scale. So we developed a multifunctional cross-linker called azide-A DSBSO or alkyne-A DSBSO. So this linker has a couple of different features. They are also targeting the lysine group, and they also have MS cleavable sites, but we added an enrichable handle used through the click chemistry conjugation for affinity purifying the cross-linked peptides. And then we have acid cleavable side to release the cross-linked peptide after affinity purification. So with this kind of linker, we generated a robust in vivo cross-linking platform. So as shown here, we can do in-cell cross-linking using the DSBSO cross-linker, and then we extract and digest the cross-linked proteins, and then we enrich the cross-linked peptides, especially the inter-cross-linked peptides, and then we subject it to the mass spec analysis to derive the interaction network and also for structure analysis and modeling. So with this information, so compared to routine proteomics analysis, you have the protein abundance expression information, and we able to generate a cross-linker proteome. And this cross-linker proteome can allow you to understand the content of the interaction, and not only with the protein information, but also their connectivity. And so we can use this XL proteome to try to do Go enrichment for understanding of their cellular functions, and also that the process is enriched by the cross-linking analysis. And then we can also compare to the known structure of all the protein complexes, then we will be able to validate the cross-links identified and also use the information to infer the unknown structure information or new interactions. And so for this identified the cross-linked peptides, we have found that most of the information generated is very close to the interaction interfaces. And then also that the protein-protein interaction captured, they have almost, they have very high string scores compared to the data present in the string databases. So with the cross-linking information, then we can derive the experimentally determined the interaction network as shown here. So these are for a couple thousand interactions, among that 2,500 nodes, and that every edges is determined by cross-linking peptides. And in addition to a global view of the proteome network, we can also define the compartment network because that protein-protein interactions can be the spatially controlled. So as shown here, that from our data, more than 95% of the interactions actually belong to the proteins within the same compartment. And in addition, we can zoom in to identify the cross-link, the interaction network in specific pathways. For example, here showing the ubiquitin proteasome system, and this is a 26S proteasome, and also its interactors and the regulators, as well as the ubiquitin machinery. So this will allow you to give you a global view to see how the network changes under different conditions when you do system-wise proteome analysis. So through the years, we actually have identified more than a dozen, developed more than a dozen sulfoxide-containing MS-cleavable cross-linker. And the first one, as I said, is a DSSO cross-linker. And then we developed this membrane-permeable, enrichable DSP-SO cross-linker for systems-wide analysis. And we developed the isotope-coded cross-linkers for quantitative analysis. And then we also developed the different reagents I showed you earlier for targeting specific residues. So recently, we also have developed a heterobifunctional cross-linker, which targeting the lysine residue, and also this end is through photocatalyzed cross-linking, targeting the non-specific residues to allow us to expand the protein-protein interaction coverage. And then recently, we also developed another cross-linker, which is the analog of the BMSO. It also targets the cysteine residues, but these allow us to do actually proteome-wide analysis. So the strategy we have developed allow us to do different types of application. And I showed you mostly example is to study on the protein complexes, and we can also study different organelles. And in collaboration with Arun's group at UCSF, we also try to study on the surface proteome and to understand the structure of the surfacome. And also that we can do on the different intact cells. And recently, in collaboration with Hui Zhang's lab at Johns Hopkins, we started to study protein-protein interactions in PDX model and the cancer the patient samples and through the CPTAG project. And so all these different types of samples can be analyzed by cross-linking mass spectrometry, and the information generated can allow us to generate the two-dimensional cross-link maps with the site-specific information, and then to determine the interaction interfaces, and allow us to generate the global view as well as the compartment view and the pathway view of the interactions. And then recently, we started to use the information and couple with AlphaFold prediction, and then hope to generate structural models and integrate with the integrative structural modeling, and then to allow us towards the system-wide analysis. So in summary, I hope I have showed you that we have developed a series of sulfoxide-containing MS-cleavable cross-linker, and so these allow us to facilitate the detection and identification of cross-linked peptides. And this technology can be used for mapping protein interactions from in vitro, in cell, and in tissue. And also, this method allows to determine the direct interactors, interaction contacts, and the network topologies, and to allow us to elucidate architecture of the protein complexes, And through the quantitative analysis, we can define the protein interaction dynamics as well as determine the conformational states under different conditions. And we hope that this strategy will allow us to push the structural system and biology. So in addition, and I think kind of related to this specific symposium, I believe that our strategy can be coupled with different affinity purification, especially proximity labeling, try to capture the E3 ligases, for the target substrates, and then to work with molecular glue, and then to identify the interactors. So at the end, I would like to thank the people in my lab. So here, the Clinton, Sharon, and Fenglong and Sean, they have contributed to the development of cross-linking technology. And several of my previous members, they contribute significantly to the technologies I described today. And my longtime collaborator, Scott Riknowski, for developing and synthesizing the crosslinker, and Ning Zhen for the long-time collaboration for the COP9 and the E3 ligases work, and Professor Burlingame's lab for support of protein prospector, Andre Sali's lab for the integrated structural modeling, and the funding agency. And thank you so much for your attention. I'd be happy to answer any questions.